Thank you so much, David, and thank you, Ran, for inviting me to give a talk at this seminar series. This is a really exciting opportunity, and then I'm really looking forward to a lot of discussions after the talk during the Q&A sessions. So as David mentioned, today I'm going to talk about unraveling information processing through functional networks. And in general, I am interested in understanding both how anatomical or physical connectivity of the neurons or neural populations can shape the population dynamics at the network level, and also the other way as well, which I'll be talking about basically starting from the population recording, the population spiking activity, how we can infer the functional networks, and then how that functional networks can actually inform us about information processing across the neural population. All right, so this is the direction that I am gonna go in. So perception, cognition, and behavior of animals induce this characteristic spiking activities of neurons. And from that neural dynamics, we can infer the interactions between neurons based on their correlated activities. So if you have spiking data from population of neurons, then by looking into the timing of the spikes, you can infer the directionality of the information flow between different neurons, or you can also think about the neural population as well. And that's how we can build the functional networks or functional connectivity of neurons or neural populations. And then because the functional connectivity by definition is constructed based on the correlated neural activity from population, it can reflect the dynamic modes of computation shaped by either the task, behavioral task, or sensory inputs, or some other internal states of the brain. All right, so what we are interested in here is basically understanding how the information is communicated and processed across the neural population by looking into the functional networks, which are built based on their correlated activities. Of course, one of the most common approaches to understand population coding of sensory information or any other information, like motor outputs or behavioral states, is by studying the population dynamics and spiking statistics. But then here we are going to discuss a slightly different approach of analyzing how neural populations compute and communicate information by looking into the network structure of their functional networks. And because the functional networks are constructed simply based on the correlated activities of neurons, looking into these topological features of these networks can inform us on understanding the information propagation across the network. All right, so there have been a lot of different methods proposed to infer the functional connectivity based on neural activities, whether it's of spiking activities or cosm activities and or fMRI and so on. So most of these methods are based on the correlations of these activities. So some simple method will be basically just looking into the Pearson correlation of the neural dynamics, or you can look into cross-correlograms and look for the sharp peaks, which I'll discuss a little later. Or you can look into transfer entropy, mutual information, grains or causality, all these things based on the information propagation between different neurons. Or another common and popular approach is to understand the functional networks of the neurons is this generalized linear model based methods like GLM methods. And of course, there are a lot more methods that have been developed in the past to infer the connectivity, the functional connectivity from neural activities. So recently, this GLM-based approaches have been getting a lot of attention mainly due to their ability to better uncover the underlying anatomical connectivity based on the correlated spike trains. So for example, this paper by Mishenko et al. From 2011, they look into they basically extract the spike timing from a population recording, and then use the GLM-based approach to infer the connectivity between these neurons based on the spikes. We also recently developed a novel one-hot map model for constructing functional networks for switching brain states. This was in collaboration with Anchiu's lab here at Georgia Tech. And then the switching brain states were inferred based on the hidden Markov model, based on from the population's spiking activity. And for each state, we can actually generate the functional connectivity between different neurons based on the generalized linear model, which is actually further informed by the prior the anatomical connectivity. Okay, and then this method was better at recovering the real anatomical connectivity in simulated spiking data, and also it achieved a higher log likelihood when applied to experimental spiking data. So, for example, in this example where we applied our method, this is from the population spiking activity from mouse barrel cortex during the whisking behavior. And the data was provided by Chris Rogers from Emory University. And then here, for our method could uncover two different internal states that are switching. And then for each state, we can uncover both the adjacency matrix, which correspond to the anatomical connectivity, which has directionality, and also the sign, basically whether it's excitatory or inhibitory, and then corresponding weight, the connection weights for the adjacency matrix. And then our uncovered functional connectivity. So this is because this was applied to the real data. We don't really know what the real underlying physical connectivity of these neurons are, but we can at least measure the log likelihood based on the spiking activity. And then when we compared our method, which is this OHG, our one-hot model, one-hot GLM model, compared to other similar GLM-based model, our method achieved the higher normalized log likelihood compared to other existing models. So all this method, so the GLM is good at inferring probably the physical connectivity underlying the correlated neural spikes compared to many other methods that we've talked about. However, in this talk, our goal is actually not about using functional networks to predict what the underlying physical connectivity looks like. In fact, inferring the real physical connectivity simply based on the correlated neural activity is extremely difficult, and it's probably almost impossible. And then there are multiple factors that contribute to such difficulty. So, for example, there are a lot of hidden neurons that you do not actually get direct measurements from, so that can actually affect your inference. And then there can be model mismatches. So, for example, for example, you can assume that the spikes are generated based on Poisson generator, but then maybe in real life the neurons do not generate spikes based on Poisson spikes. So the model mismatch can also hinder the correct inference of the physical connectivity based on correlated neural activity. However, what we are interested in here is not really about uncovering the underlying anatomical connectivity that can based on the correlated neural activity. Rather, our focus here in this talk is on understanding the information processing across a neural population through the lens of functional networks. So it's a slightly different approach to use functional networks. So, all right. So, one interesting feature of visual, in light of doing that, I am going to introduce different types of visual processing that we can understand by looking into the functional networks. And first of all, we are going to look into the functional network structures constructed from CCG or cross-parallelogram applied to this neural pixel recordings from the mouse visual cortex during different visual stimulus presentations. And then, so this is applied to this data set from the Allen Institute, obtained from electrophysiology data by using neuropixel from mouse visual cortex and thalamic areas. So there are this. So these are all visual thalamus and cortical areas. And then the spiking activities were recorded based on neuropixels during different stimulus conditions. So, for example, there is this spiking data from the resting states, flashes, drifting gratings, static gratings, natural scenes, and natural movies as well. And just to get, and then because this is a huge data set, and then it contains a lot of neurons, it's actually a little hard to use GLM-based methods to uncover the functional networks based on this such a large data set. So what we used here is the cross-correlogram-based methods, and then we use the cross-correlogram-based methods to understand first the functional hierarchy of the visual cortical regions. All right, so in this 2021 paper, correlation structure between neurons in mouse visual cortex and thalamus were analyzed based on this large-scale neuropixel data. In this paper, we only used the drifting grading, just one type of very simple stimulus, to understand the functional hierarchy of the visual cortical areas. So during the drifting grading stimulus, the recordings were made from these different visual areas. And then by using the cross-parallelogram, we identified a sharp peak, which indicates the directed connection, functional connection from one neuron to the other. And we looked into the distribution of the CCG peak time lags between each pair of the visual areas. So for example, this shows the distribution of the CCG peak time lags between V1, the primary visual cortex, and LM. And then you can actually see the overall directionality of the connection from V1 and LM. And then based on this distribution of the CCG peak time lags for each pair of the regions, you can build this directionally sparse matrix for source regions and target regions. And then this will give us inform us on the hierarchy of these visual areas. So here, what I mean by hierarchy is just the directionality of the information flow. And what was very interesting here is that there is a good correlation between this directionally score that we found from the cross-correlogram peak times and the anatomical hierarchy score difference. All right, so this is the anatomical hierarchy of different visual regions, different mouse visual cortical regions obtained from the anatomical connectivity data. Which I'm not going to go into too far. And then when you look into the anatomical hierarchy score, then the difference between this difference in this hierarchy scores between pair of regions, and then if you look into if you compare that to the directionality scores based on the CCG peak time lags based from the spiking data, we have this good correlation, which indicates that the functional hierarchy obtained from this electrophysiology, actually during only drifting gratings, agrees very well with the anatomical hierarchy. Okay. However, as I mentioned, the one limitation here is that the functional hierarchy was obtained only during one stimulus type, basically drifting grading. So it just gives us some limited information about the functional hierarchy, and then this is the functional hierarchy obtained only when there is a very simple visual stimulus. So, for example, if you have a different or more complex visual stimuli, then maybe the hierarchy changes. And then that's actually a very cool thing about the functional hierarchy, because unlike anatomical hierarchy, which is basically fixed and it never changes, functional hierarchy can change depending on the stimulus types or behavioral states and so on, because these are dynamically changing networks based on the correlated neural activities. So therefore, our next question is to understand how the stimulus complexity can affect the topological features of the functional networks of neurons. So the main question that we are interested in here is basically whether the complexity of the functional connectivity or the functional network is shaped mostly by the complexity of the input visual stimuli, or is it more dependent on the underlying anatomical connectivity? So what defines the complex structure of the functional networks network structure? So to understand this, we looked into the spiking data from this visual cortical areas from mouse during different stimulus types, rather than just focusing on the drifting grading. So this is our recent work led by a very talented student, Disheng, where he looked into the Allen Institute's neural pixel data set and then divided this spiking data set into different categories based on the stimulus types. With different complexity. So, for example, like resting state and flashes are relatively simple. It should be simple, relatively simple visual stimuli. And same with drifting gratings and static gratings, that those are relatively simple visual stimuli. But then we assume that natural scenes and natural movies are more complex visual stimuli. And then we were interested in how the emergent functional networks of the neurons from this visual cortical areas can be shaped by the stimulus complexity. So in order to do so, Disheng basically built the functional connectivity of single neurons across all different visual areas that correspond to each visual stimulus type. And then this is how he did that. So first of all, so we have the CCG between CCG based on this based on the spiking data, and then the CCG was jitter corrected. So the jitter corrected CCG was created by subtracting the expected value of CCGs produced from the resampled version of the original data set with spike times randomly that were randomly perturbed within the jitter window. So that the jitter correction can remove the slow time scale of the correlation larger than the jitter window. So So this jitter correction method can remove both the stimulus lock component of the response as well as the slow fluctuation larger than the jitter window. And then it can give us the better picture of the interaction between single neurons. And then we identify the sharp peak in the CCG, so that we are interested more in the direct interaction between single neurons rather than the polysynaptic interactions. And in our method could also identify the excitatory and inhibitory connections based on their peak and trough. And this is the initial CCG-based functional network at the single cell level across all different visual areas. And then we thresholded it so that it's going to be a lot more sparser. All right. So what is not nice about the CCG-based method for developing a functional network is that, first of all, it's relatively fast and it can actually deal with this large data set with a lot of neurons. And also we can, and then our functional network is both directed and then it has sign, basically excitatory and inhibitory. So this gives some examples. So here, when there is a sharp peak in the CCG, then you can define that there is a connection. And then depending on whether the peak occurs after 0 or before 0, you can see the directionality of the edge from one neuron to the other. So if you see 2 peaks, then it's a mutual connection. And then depending on the sign of the peak compared to the mean of the CCG, you can define whether it's excitatory connection when it's positive or inhibitory connection when it's negative. And in order to understand the network properties of the CCG-based functional network of single neurons in the visual cortex, we need some sort of surrogate network that we can, or the reference network that we can compare with, right? So as surrogate networks, we tested a few different models. So first of all, Erdős-Rényi model, which preserves basically just the number of nodes, which is V, and the number of edges, which is this E here. Or we can compare the real functional network to degree preserving model, where, as the name says, the in degree and out degree of the networks are preserved, while other features are perturbed. We can also look into the pair preserving models, where the neuron pair distribution is preserved. And finally, we can also use the most conservative reference network, which is the sign pair preserving model, which preserves not only the number of edges and number of nodes and edges or degrees, but then it also preserves the neuron pair distribution that actually has the signs, whether it's excitatory or inhibitory. And then we can compare the network properties of the real network generated from the experimental data, and then compare that to the network features of the surrogate, the reference network. And then in most of our analysis, we use the sign pair preserving model as our reference network because this is the most conservative reference network. And then so there are several different network features that we were interested in to study from our functional network. So the first feature was this low degree motif structure. So here, this shows the three neural motifs that we found from the functional connections. Of course, there can be a lot more different three neural motifs from the functional networks. But then this, and we were interested in this specific class of three neural motifs, which is called the feed-forward loop. Which has this like feed-forward features that actually propagates from like one neuron to other. And then because we are dealing with signed the signed network, where we have the excitatory and inhibitory connections, we can actually further divide this feed-forward loop, which is this one, the feed-forward loop, into the signed feed-forward motifs. Okay, and then yes, signed feed-forward motifs based on the sign of the edges. And then this class of three neural motifs, we call them excitatory feed-forward based motifs. And then why these motifs are interesting, that we are gonna see next. All right, so this is why these excitatory feed-forward based motifs are very interesting. So here, this shows the motif intensity significance compared to the sign pair preserving surrogate networks. So this is basically the interesting feature of the functional network constructed from the experimental data. And then what we see here is that so each row represents different stimulus type. So the first one, so the bottom one is the resting state. This one, I don't know whether you can see my cursor, and then the second to the bottom one shows the flashes, drifting grading, static grading, and natural scenes and natural movies. And what was very interesting here is that across all different stimulus types, we see that this excitatory feed-forward loop motifs are always overrepresented. And what was actually really interesting, so the next question that we're asking is, so it seems like these motifs are always overrepresented, but then does that mean that it's basically the same neurons that constitute this three neuron motifs all the time throughout all different stimulus types? And the answer is no. So here in this figure, it actually shows that the intersection of unique motif set during this, well, so this actually shows the intersection size of the unique motif sets with different constituent neurons for different stimulus types. So here, this is natural movie, natural scenes, drifting grading, static grading, flashes, and resting state. And what we see here is that the intersection is what we are seeing here is that a large number of unique motifs appear only during one type of stimuli. And then basically that demonstrates that the component neurons are always changing. So it's not the same neurons that are present across different functional networks. So the consistent overrepresentation of this excitatory feed-forward loop-based motifs suggests that they can be a key information processing subcomponents of the neural circuits. So some speculations of how this excitatory feed-forward loop-based motifs may be a good functional, can actually work as a computational unit is following. So for example, one of the excitatory feed-forward loop-based motifs can actually work as a sign-sensitive filter that responds only to persistent stimuli in transcriptional regulation networks. So of course, this is not direct applications to neural networks, but then it can actually have some computational feature. And also multi-input feed-forward loop is found to store memory and reject transient input fluctuations in neural networks. So while we don't really know the exact computational advantage of having this excitatory feed-forward loop-based motifs across all different stimulus types, it may actually serve some computational role, or it can be as just some emergent properties. We don't know that. And we also looked into a large-scale modular structure of the network as well. And then we found that the modular structure changes with stimulus complexity, unlike the three neuron motifs, which is actually basically preserved across all different stimulus types. And by using this adjusted RAND index, which is a network level measure used to quantify the similarity between module partition and the visual area organization. So basically, this measure is higher if the modules of the network correspond, like aligns very well with the actual cortical regions. For example, like V1, LM, AM, and so on. And then what we see here is that this measure, the ARR, adjusted RAND index, increases with the stimulus complexity, roughly, which means that so here, this is natural movie and natural scenes, drifting grading, and static grading. And then what this basically tells us is that as the stimulus and complexity increases, it seems like the functional modules are more contained within the visual cortical region, and more spatially segregated. So and then that actually suggests that the functional modules with more spatially segregated structure could be required in more demanding cognitive tasks. All right. So that gives us some ideas of how different stimulus complexity can be reflected in the functional network structure of the single neurons from the visual cortex. And then the next question that we are interested in is to understand how the functional networks can inform us on the neural processing of stimulus novelty and unexpectedness. All right, so surprisal or novelty captures our attention, and it can also enhance learning and memory as well. So say you go to a museum and then you're in an exhibition room of Dutch painters. And as you walk by, you're passing by a bunch of portraits of a similar style. And so when there is some unexpected paintings of a strikingly different style from a different era, then you're definitely going to notice that, and then you're going to remember that better. So here we ask the question, how the information about the surprisal and novelty are communicated across the neural networks by inferring the functional networks. All right, so to do so, we focused on this two-photon data set also from the Allen Institute, which you can also find in this paper by Marina Garrett et al. So in this study, the mouse was trained to perform a change detection task where it's presented with a repeated natural images. So for example, flower, 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 and flower. And then whenever it's presented with an image change, like right here, then the mouse has to click to receive a water reward. And then furthermore, the images presented in sequence can be selected from either a set of familiar images that mouse has seen a lot, or from a set of novel images, which mouse has never seen. So, and then while the mouse was doing this visual behavior task, basically the change detection task, the calcium, so the two-photon image recordings were made from two different visual cortical areas, namely V1 and the higher visual area LM. And then the recordings were made across multiple depths in the cortical depth, so that we can actually relate the neural activities to the cortical layers. And also, we could actually get they could actually get this cell type specific response patterns to image changes, familiar images, and novel images. Namely, they made recordings from excitatory neurons, somatostatin SST inhibitor neurons, and VIP inhibitor neurons, which are known to have this typical connection patterns. So, so while the animals were engaged in this visual detection task, so calcium activities were obtained from this different cell type across different depth and across two different visual areas. And they found that different cell types could actually exhibit distinct response patterns to familiar and novel images, and also to the image changes. So for example, their recordings from the excitatory neuron shows that it responds strongly at the image change. So here the gray bar represents the last repeated image, and then the blue bar represents the change of the image. So it's like flower, 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 and cat here. And then when there is image change, then the excitatory neuron responds more strongly. And overall, the response was higher when the images were chosen from the novel set of images that the mouse has never seen. And then when the recordings from the VIP inhibitory neurons actually show the very similar response trends to the excitatory neurons. However, the response trend was very different in the SST inhibitory neurons. So here, what they found here is that for the familiar, when the images were chosen from the familiar set of images, then the SST inhibitory neurons respond more strongly compared to the images from the novel set of images. And then in this paper by Garrett et al., they speculated that the stimulus novelty basically engages this VIP, SST, this inhibitory circuits. So the excitatory neurons, which has this response pattern, has the same response pattern as the VIP inhibitory neurons, which inhibits the SST neurons. And therefore the SST neuron has a very different response patterns here. All right, so so this was a very interesting data set showing that different cell types across different brain areas across the cortical layers can have different response patterns to novelty or unexpectedness, temporal unexpectedness. And then the next question that we were interested in is, okay, so can we actually infer the information flow or the directionality of the information flow across different cell populations and different brain regions based on the functional networks? All right. So in this work, previous work by Parrot and Rajan, they developed this method of inferring the information flow between multiple brain regions by using recurrent neural networks. So what they did here is to correspond different RNN to different brain regions and basically feeding the responses from the RNN to the data. So basically the time series data from the brain regions. And by fitting up, and then the learned connection weights between different RNNs representing different brain region can actually denote the functional connectivity or functional networks of these different brain regions. And then this is the method they call curve. Curve to infer the information flows between different brain regions. So we were inspired by these methods, but then this methods has some limitations if we want to apply that to uncover interaction between different cell types and layers in the cortical microcircuits that we want to use to understand the information flow during the visual behavior task from the Allen Institute. Because first of all, the conventional RNN training does not preserve the sign of the projection weights, and therefore it violates the Dale's law, right? So, secondly, the conventional RNN training does not constrain on anatomically informed sparse connectivity patterns across different cell types, layers, and areas that we are informed by the experiments, while also maintaining good training performance. So these are two limitations that we identified. So we implemented some modifications to this RNN-based approach to infer the functional connectivity between different neural populations that address these two problems. So this work was led by a grad student, Aish, in my group, and also Alex, also a grad student in my group. Aish and Alex tried to address these two limitations. So the first limitation was about the method, the RNN training method, not conforming to Dale's law, right? So in our modified model, we can actually train the network with sign preserving back propagations. So how we did that is basically to initialize the recurrent connectivity matrix to correct signs for inhibitory and excitatory projections at the very beginning. And then at each time step, we did this sign preserving back prop where we have this regular back prop, and then we have this additional step P sub C, which keeps only the components that have the same sign as the previous time steps. So overall, we can actually learn we can actually train the RNN while preserving the signs of the connections. Okay, so and then there was the second limitation where like in the conventional RNN training, we cannot really do a good you cannot actually really achieve a good performance when you are enforcing this anatomical sparse connectivity that are informed by the experiments. So in order to address this, I use this post-training pruning method to match the data-informed connection sparsity for each cell type and layer-specific connection projections. And the connections are pruned to achieve a target sparsity where each edge is preserved with the increasing probability with its weight. And then after this post-training pruning, the weights are trained once again. So there was a secondary pruning, so to make the performance better. And we tested whether this two modification could actually achieve a good performance in RNN while preserving this like Dale's law and also the sparse anatomical connectivity. All right. So this is our simulations with the sign preserving back prop when it's applied to RNN to perform this wave reconstruction task. And what we found here is that compared to the conventional backprop, our method, which is this blue line here, performs pretty well in terms of the test losses. And then it actually performs better than some other backpropagation methods as well. While preserving this the excitatory and inhibitory signs for the projections. And then our pruning method indeed preserves the test performance while maintaining this anatomical connectivity sparsity informed by the experiments. So this is also from the same wave reconstruction task. And then here, this is what we what we see here is that so the green dots here represents the errors from errors from our pruning methods compared to the random pruning methods. And then what we see here is that our pruning method, after the secondary training, it actually does really well in terms of the wave reconstruction task. So the data-driven target connectivity sparsity is preserved in the pruned weights, while the RNN can still do very good performance in wave reconstruction. All right, then, so now we have these two modifications in the RNN-based method to infer the functional connectivity. We now apply this method to the Allen Institute's visual behavior data to uncover the interactions across the visual cortex during this visual behavior. So, to do so, we first started with this RNN model of V1 and LM circuit, where it actually has this canonical cortical microcircuit between different layers and across different areas as well. And within each cortical layer, we have different cell types, namely the excitatory neurons, SST neurons, and VIP neurons. And then we want to actually preserve the connection sparsity informed by the experimental data for each cell types across different layers and across visual areas as well. Okay, so similar to the curve method, what we are doing here is to try to reconstruct the data, so the time series data of the neural responses from different cell populations across different layers, cell types, and areas. So for example, and then try to look into the learned connection weights, which will represent the functional connectivity between different cell populations. And then we did that for different types. So for example, when there is no change in the image, when there is a change in the image, or and then when these images were selected from the familiar set of images or novel set of images corresponding to different experimental sessions. So here, this shows an example. So this is an example. So the dotted line here is the actual experimental data showing the average response pattern, average response pattern during the image change. So here, the dark yellow shows the previous image, and then the bright yellow shows the change of the image. And then, so this is how the VIP neurons in layer 5 of the LM response in this case. And then what we did is basically trying to train our recurrent neural network so that the subpopulation that represents this VIP neurons in layer 5 of the LM matches this experimental data as closely as possible. Okay. All right, then. So because we are doing that for different cases where there is expectation violation or where there is novelty, what we wanted to understand was whether the functional connectivity that was uncovered based on our method can explain predictive coding theory. So predictive coding is a theory of this hierarchical cortical computation where it postulates that the brain actually maintains this internal representation of the world, which is actively compared and updated based on the incoming sensory inputs. And feedback signals are speculated to carry predictions. Sorry, feedback signals are speculated to carry predictions about the lower area activity, while the feedforward signals represents the residual errors between the feedback predictions and the encoding expectation. And in fact, our method uncovers this functional interaction between different cell population that actually kind of agrees with the predictive coding theory. So here, this is what we found. So here, this shows the functional connectivity based on the learned RNN weights during learned RNN weight. Actually, this is a difference between this learned weights between the no change condition where the images are from the familiar set of images and the change condition where the images are from the familiar images. And then this basically shows that that there are some enhanced feed-forward projection coming from the V1 layer 2-3 to LM layer 4 when there is an image change, which can actually represent the signaling of the prediction errors. And then there is this enhanced connection from the layer 5 of the LM to the layer 2-3 and layer 5 of V1, which may represent the feedback predictions. We also noticed that the VIP inner neurons exhibited increased connectivity during the change conditions in both V1 and LM, layer 2-3 and layer 4, as you can see here in this magenta box. And then this may actually correspond to processing of the unexpected input, which is this change of images. When we can also just look into so that was actually these connections were inferred when we are looking into the full session from the previous image and the second image at the image changed. But then we can only look into this like short period of time, like right after the image change or the repeated image in the second part, right? And then what we see here is that okay, so we still see this enhanced feed-forward projection from layer 2-3 of the V1 to layer 4 of the LM when there is an image change. But then we see this reduced feedback from LM to V1 when the period was shortened. Then this may actually indicate that the propagating feedback information can occur over a longer time scale. And finally, this actually shows the connectivity difference between familiar no change condition and novel no change condition, where we see this enhanced feed-forward projections from V1 L2-3 to LM L4, which when there was a novel node change compared to the familiar node change, which may actually signal the prediction errors as well. Okay. So with that, to summarize, the functional connectivity can be inferred by various methods, as we just discussed, and the functional networks of mouse visual cortex shows hierarchical structures. And the network topology of the functional connectivity of neurons inferred by the CCG method, we see that the topology is driven by the stimulus complexity. So first of all, if you look into the local structure represented by the 3 neural motifs, we see that there is some preserved overrepresentation of the excitatory feed-forward loop motifs across different stimulus types, which actually suggests that it may actually serve as a local computational unit. And then the stimulus complexity induces the functional modules with more spatially segregated structures. And also our modified RNN training method can uncover the transmission of predicted and unexpected visual information across the visual cortical circuits. Okay. And then with that, I would like to acknowledge my collaborators, students, and my funding sources. Thank you.